and welcome to another episode of Travels Through the Body. Today we will be talking to glucose about its recent trip through the kidneys. Come on down, glucose. Go.
All right.
Let's talk about your trip south. Everyone is buzzing to hear about what's going on in the kidney. What is it like this time of year? Well, I can tell you around the holidays is not the best time to go. The adrenal glands, which are superior to the kidney, produce way too many stress hormones like cortisol for my taste. Did you meet anyone special? I did spend some time with an interesting albumin as we traveled through the initial arteries of the cortical nephron, but we were separated at the glomerulus because he was too big to fit through the filtration slits. So you traveled the renal tubule alone? Well, I didn't make it through the entire tubule, or I would be in the toilet rather than talking to you. Okay, well then what parts did you travel? From the beginning? From the beginning. Okay, well, I took the aorta to the renal artery. Wait, you use public transportation? Oh yes, I think it keeps me in touch with the body. We all have to work together to achieve and maintain homeostasis. Anyway, we got to the kidney and chose a cortical nephron. We took the segmental artery to the interlobular artery, to the arcuate artery, to the cortical radiate artery, to the afferent arteriole, and finally to the glomerulus. I was really looking forward to seeing the foot processes of the podocytes, but there was way too much hydrostatic pressure, so I moved through the filtration slits and into the glomerular capsule. So you spent some time in the filtrate. What did you see there? It actually looks a lot like plasma minus the proteins. There were vitamins, minerals, amino acids, pretty much anything petite like me. The glomerulus isn't too judgmental, except about size. From what I've seen of the renal tubule, it is much more selective. But I've only seen the initial portion, the proximal convoluted tubule. What did you experience in the proximal convoluted tubule? There is a dense forest of microvilli for surface area and a ton of mitochondria. Which reminds me, I want to shout out to the enzyme sodium-potassium ATPase, also known as the sodium-potassium pump. Without them to regulate the electrochemical gradient, I never would have made it out of there. Interesting. Tell us more about the process of primary active transport. You mentioned that this is the process that drives reabsorption for nutrients such as yourself. They drive the process of reabsorption by a process called primary active transport. They bind 3 cytoplasmic sodium molecules, which promotes hydrolysis of ATP to ADP. This phosphorylates and causes it to change shape, sending sodium to the outside, where 2 extracellular potassium molecules bind, triggering the release of the phosphate. The protein resumes its original shape. When it binds ATP again, it releases potassium into the cell. This activity creates a strong electrochemical gradient that enables me to enter the proximal convoluted tubule cell along with sodium through a SIM port, which is a sort of VIP door for only me and sodium. This process that I use is called secondary active transport. Once I've been escorted by sodium through our SIM port, I just go with the osmotic flow from the tubule cell all the way to the peritubular capillary. Which reminds me, I really need to get back to work. You know, the brain needs me for energy. It's very particular. Well, alright, there you have it, from filtration at the glomerulus to reabsorption at the proximal convoluted tubule. Thank you for joining us. Good night, folks.